For those of you who have joined us, my name is Jim Bernau. I am the founder um, and CEO of Willamette Valley Vineyards. I want to thank you very much for joining me. They say that wine is drinkable history. And that's really one of the things that's really how I'm approaching this. Here you're seeing, for example, um, the vineyard that I first planted when I started. I found the property in 1981 and began planting it in 1983. And the, there's a lot of science that goes into selecting a site. The soil, the weather conditions, the general climatic conditions, orientation to the sun, elevation, a whole variety of factors that make it an ideal site. But like in many parts of life, there's these aha moments. And my aha moment for selecting this site was, despite all of the science and the detail behind it, was when I knocked on the neighbor's door down at the bottom of the hill and asked them about whether I should buy this property or not. They said, "Oh, Mr. Bernau, I wouldn't buy that property. My relatives farmed that property many years ago, planted plums, which they dried into prunes that they sold to gold rushers in California." And the lady said, the problem is that as the plum trees went up the hill, she said the plums got smaller and smaller, and when they dried them, they got much less money for them when they shipped them. And of course, my heart was pounding when she told me that, when she said that the plums at the top of the hill, they described as whiskey prunes. And so it's kind of an aha moment. There's some other aha moments in the story of clones. Now, when this issue of selection of clones started, it was really about looking at in this next slide, you'll see, looking at this cluster of Pinot Noir. The physical characteristics is really what clones were initially selected upon. Were they tight bunches? Were they loose bunches? Were the vines grew upright, or did they droop? Which all have kind of wine growing significance. And then of course later they were tested for their aroma and their flavor. But these characteristics are the things that we're going to cover today, and why that is important to us. Of course, Pinot Noir means pine, like pine cone shaped black, and you can see that Pinot Noir is is actually a tightly clustered grape shape that's cone shaped. Here, you might not see this as tightly spaced as you see it. And so what I'm going to do is I'm going to offer this to any one of the people here. If you, by the end of the night, send to me a description of what Pinot Noir clone this is, provide as much description as you can. You don't have to be precise as to what the clone is, but why you believe it's a particular clone of Pinot Noir by looking at it. So you might take a moment, take a picture of this slide, so you can maybe refer to it later to see if you can win 6 bottles of our best Pinot Noir by being the person closest to what this Pinot Noir clone is. So that contest is open through the end of this evening. Now, when clones are described, they're described by location or by town, by the vineyard owner, or the name of the vineyard, or the ranch, like in California, or by numbers. And even these numbers change over time. So this is really, it gets pretty confusing. But anyway, if the way to contact me on your answer is my email address, Jim, Jim.Bernau, Bernau, @wvv.com. And you can send me your best guess as to what clone of Pinot Noir this is, and the answer will be hidden in this presentation. So hopefully you'll hang around through the presentation, because it's really intended for the staff, and so it's really fairly comprehensive. I have 45 slides, and I'm going to move as fast as I can. The way I learned how to pronounce Dijon was when my brother got a yellow lab before he went to college. When he left high school, he, you know, as you probably know our history, my brother and I dabbled in winemaking when we were young because my dad got us inspired. My dad was hired by Richard Sommer, Oregon's very first vinifera winemaker, to come to Oregon. And my dad got us excited about what Richard Sommer was doing. I was 10 and my brother was 12 at the time when we got to taste Richard Sommer's wine at the dinner table. Well, my brother really got the bug. He was 2 years older than I was. So in 1969, after he graduated, he went to the University of Dijon to study winemaking. And he left his dog with me, Dijon, that he named after the university. Now, one of the things about clones is that, just like these hydrangeas, you'll see in the next slide, is you've got to keep this all in perspective, because Pinot Noir or vinifera vines are just like these hydrangeas. These hydrangeas are actually genetically identical. But as you can see, they have different colored flowers, and you're going to see why in this chart by looking at their pH. You see the soil pH, just the level of attraction that exists, the electrical attraction that exists in the soil, those positively charged hydrogen ions, the more there are, so the lower the pH, that's the higher concentration. So it's kind of a reverse measurement. The higher the pH, the lower the number of positively charged electrons that are in the soil. And so what that means is that when you change just the pH in the soil, the color of those flowers change. And this is called an epigenetic characteristic. And hydrangeas have it, as well as vinifera and Pinot Noir have it. So you have to keep these clones in perspective, because there's a lot of things can influence how the wines taste. For example, a particular clone 667 grown in soil that's dry, that's shallow, like it is at the estate, has a beautiful kind of holiday spice, or what we call Pinot Noir spice in the aroma. Whereas if it's grown in a very deep soil that's very vigorous, it doesn't taste very good at all. And so this will give you at least a little idea to keep these things in perspective. Remember, most of this plant material now is grafted onto rootstocks because of phylloxera. So there's this phylloxera-resistant rootstock that actually is in contact with the soil. So that's a whole nother story, is the influence of rootstock on how the wines will, their aroma and their taste. Now, we are launching our very first Dijon Clone Pinot Noir, the 2019 vintage, and I wanted you to tell you that you know, you think about, you know, kind of barbecued salmon, for example, but for me, I'm reminded of when my now wife Jan made a dish for me when we were dating, called chicken marbella. And I think that's probably one of the first nights I felt like, well, I was really falling in love with her. She made an incredible chicken marbella. And this wine would be a perfect match with that. We only made like 50 barrels of this wine. It's all for these Dijon clones. And when you look at the back label, you'll be able to see those clone numbers in the technical data on the back label of this wine. Man, it's, you know, I hope that for those of you who have it, you're putting it up to your nose, you're tasting it, and realizing that it has these wonderful kind of red cherry, raspberry, and also kind of a little kind of red clay earth, you know, from the soil. It's juicy. It's kind of got some sweet barrel toast and very vibrant. So this is a new offering we're just launching to feature the Dijon Clones Pinot Noirs on its own. And the second wine I wanted to show you was the Chardonnay, which of course is many of you have seen, because this has been actually available for some time now. And this is made up of, when you look at the back label, it's made up of it's actually principally made of two clones, the 7.6 and the 96. I did tell my staff we actually have a little 95 planted in a row of that 96, because that's all I could get. And they didn't know that. But anyway, it's predominantly the 76 clone. And just think about when I was in college, I made a lot of chicken pot pies, but actually they were frozen. But if you are up to making a chicken pot pie, make a chicken pot pie with this wine. It's got kind of this lemon pear, just kind of a honeyed aroma. It's got sweet cream in it, maybe a little vanilla, citric orange. Anyway, all these clones really, when put together in the combination they are, really make a wonderful food companion. So how did this all start? So this is how it started. It started with the problem of a virus, just like the problems we're having today. This virus, leaf roll, actually plagued the growers in Europe. So in the 30s and 40s and 50s, what happened was this leaf roll virus started getting into the vineyards. It's transported by insects or by touch. And what it did was it drove down the yields of their vineyards by 50%. And it actually affected the color in the grapes. A virus did. And so the winemakers in all of Europe, but in France and in Burgundy, asked for help from the French government to ask them what they could do about it. And here's who came to the rescue: a Dr. Raymond Bernard, and he's a remarkable individual in the whole story of saving the wine industry in France and in Burgundy. Here you can see that in the picture, the University of Bourgogne, and that's what the French actually call Burgundy, and that's actually how they prefer us to refer to it. But I've been calling it Burgundy for a long time, but it's the University of Bourgogne in Bourgogne. And this is where Dr. Raymond Bernard started as a researcher, then became a professor, worked his way up. And when he started, he really had the task of figuring out what to do about this leaf roll problem. And he did, he figured it out. But in the process of him selecting plant material that was free from this virus, he had an aha moment. And he thought, oh my gosh, what if I also separate out these clones for other benefits or other advantages? And so by looking at the clones visually, looking at them, looking at the vine and how they grew on the vine, he created a series of classifications by number. Now, these numbers are just simply the ascension of these particular clones being added to their system. So there's no significance on these numbers. But here's the problem that Dr. Raymond Bernard had. He had to deal with the tradition of the French winemakers, because they grew their vines in what they call a Masala Selection. And a Masala Selection means that the wine growers would pick the very best vines in their vineyard, which were genetically very diverse, and then they would continue to increase their plantings or replace their plantings with these best cuttings. Well, and so what Dr. Raymond Bernard was saying was, well, let's separate this out as clones, as distinctively different clones. And actually the growers there and the winemakers were not enthused about that. And so when he first started, he used his own money. Think about the very meager salary he had as a young professor. He had to use his own money to start this process. But that was the aha moment that made it possible for winemakers around the world to do something very unique. This next slide will show you one of our leader actually in the vineyard, Efren Loaiza, and he's got cuttings in his hand. I wanted to make sure you saw what they were. And he takes them, as you can see in his hand on the left, he's cutting them off from the growing shoot, which is turned into a cane, a 1-year-old wood, over the winter. And that's what that's his cuttings that he is then going to use to plant in another vineyard called Loaiza Vineyard. But now here's the thing I wanted to make sure you understood. Here's the problem with this. The problem is that if there's viruses in that plant material that are damaging to the vine or to the fruit, you've got a problem. So you could spread this virus even further. And so what the Californians did was they first tested trying to deal with viruses in potatoes in 1936. And then they hired a fellow named Professor Olmo, another one of the remarkable contributors to the wine industry. Professor Olmo, University of California, Davis, was he's really one of the reasons why we have so much opportunity in diversity in our vineyards is because of his work. Now, he figured out by using heat treatments, basically putting those cuttings in a very hot environment, basically at 100 degrees for 60 days, almost killing all life inside that cutting, he could kill the viruses. Now, what he learned later was it wasn't 100% effective against certain viruses, but at least it was a start. And that's what the technology they had at that time. But and what I think here's what they didn't know. What they didn't know at the time was not only were they killing the viruses, they were also genetically altering the plant itself. That little shoot that would come out of the bud after that heat treatment was genetically altered. And the reason why we know that now is that there's a germplasm stock, Australia, that's the name of the company in Adelaide, that has figured out by applying heat over certain periods of time, you can actually not only kill viruses and bacteria that are harmful, but you can actually genetically change the plant to require less water, to require less fertilizer, to be more fruitful, and even the timing of ripening. That's actually something that's been fairly new in discovery. So anyway, one of the things that Dr. Olmo had was he had a person working with him by the name of Dr. Cohen. And so they wanted to go and start this experimental vineyard and gather up as many of these different cultivars and different varieties and different versions of varieties, mutations of varieties as possible. And so they went in together with Louis Martini. Louis Martini, by the way, was Bill Fuller's boss. Louis Martini Winery in Napa, and they went and got some land at what's called the Stanley Ranch in the Caneros, and that's an old vineyard that escaped phylloxera because it was isolated in Caneros, planted like in the late 1800s. And this Dr. Cohen went around trying to find this plant material, right? Besides getting it from Europe, they went looking around for it from growers, and they found an old vineyard just south of Folsom, California, in a little town, a gold mining town called Jackson. And this is where the University of California planted a vineyard in 1883, and then in 1903 abandoned it and basically lost track of this vineyard. And so this vineyard was growing for 60 years, and the only persons tending it and pruning it were the deer. Anyway, this vineyard was planted originally to help the gold miners develop new skills as the gold mines played out. So isn't that strange that the Natoma vineyard was actually the plant source material for what's called the Jackson vineyard, one of the sources of these heritage clones. Now, when you look at how the vines are, what we do is when we buy vines, we have a graft. These vines have been grafted. And this is where the scion wood, the bud has been kind of cut into the phylloxera-resistant cane. And so that's actually, the phylloxera-resistant cane is actually an American rootstock because it's resistant to phylloxera. And that wax holds that little insertion in place until Mother Nature, and the vine can start growing and then fuse it together. But what happened was they discovered in this heat treatment approach that it wasn't 100% effective. And so they went to this next approach. So this is another innovation in how to produce different clones of Pinot Noir and Chardonnay. And this is called the micro shoot tip. And so you see on the far left that little piece they start with just a little teeny shaving off the top. And it's got a meristem, and then it's got 2 to 3 leaf primordia. And that's it. It's about 0.05 millimeters in width. And that's what they start. And what they found is when you do that, you don't have any vascular material around those primordia, so the viruses don't have any way of getting onto it. So that's what they found to be 100% effective in defeating these viruses. And that's what they all use now. And to make things even more complicated, the numbering system used by UC Davis in numbering the vines is that every time they go and treat a vine differently, let's say when they went from a heritage clone, in other words, a native true variety, That's kind of a heritage that hasn't been heat treated. That has a different number. Then they heat treat it, that has a different number. And then when they do the microtipping to eliminate even more viruses, that has a different number. So that's one of the reasons why I'm not giving you all these numbers, because it would drive you crazy. Now, for us in Oregon, what we did was Oregon received its first grapevines through the work of Richard Summer. Richard Summer was, as you know, he was studied at UC Davis, and he was the first student of enology and viticulture to move to bring vinifera vines into the state of Oregon, vinifera Pinot Noir vines in the state of Oregon, from UC Davis, and actually his classmates were Bill Fuller and David Lett at that time. My dad met him when he had a day job. He was the Douglas County deputy assessor, where he met my dad and asked my dad for help in getting a winery license and forming a business. And that's how my dad encountered Richard Summer, and that's how I encountered Richard Summer's wine and my interest in wine. Well, anyway, what Richard Summer did was he brought cuttings up from this partnership between UC Davis and Louis Martini at the Stanley Ranch. And he brought up Wente Clone Chardonnay, and he brought up Permard. But at the time he brought them up, there was no heat treatment going on at UC Davis, which means those vines are the real McCoy. And I don't know how many people really know that. Here's a cluster of Permard being held in your hand. As you can see, look at this cluster. See how tight those berries are put together? Well, now, this is one of the most popular, actually, this was the most popular and most planted variety, in part because Richard brought it up, but also people like Dick Erath got started a nursery with another winemaker in Oregon, Chuck Corey, which I'll talk about in a minute. And they actually really helped other wine growers get started. So they got some of this plant material coming up from UC Davis, and they got the heat treated Permard. So that is genetically, frankly, different from what Richard Summer first got. But here you can see how tightly formed, just like a pine cone, in this particular clone of Pinot Noir. Now, I want you to imagine another student at UC Davis studying. Now, this student was studying meteorology. He was going to become a scientist using math, and he was watching a movie with his wife when he was at UC Davis, and the movie was called Battleground. And this movie, and then that's in the next slide, Mandy. This movie is of World War II. It actually was released in 1949, starring Van Johnson, so this young aspiring meteorologist was watching this movie, but he wasn't quite sure if he wanted to be a meteorologist. But he was becoming basically a scientist using mathematical formulas to study the weather. And when he saw this movie, you see that sign that says Bastogne? Well, in this movie, that sign, there's a sign that says Bastogne, and then there's an arrow to a town spelled R-E-I-M-S, and that's actually pronounced Reims in French. And that is the heart of Champagne. And so the aha moment for this young meteorologist student, Chuck Corey, was, aha, I see cold weather. I see cold weather. I know that it's cold in Reims, and they have some of the best wine in the world they're growing there for sparkling wine. What? This is not what they're teaching at UC Davis. And so his light bulb moment was, and then he, of course, started focusing on proving an idea, a hypothesis. And his hypothesis was called the cold limit amelioration hypothesis. And that's what his thinking was that the best fruit from vinifera vines ripens right at the limit of the growing season. That was his theory, and he believed that it was actually the best place to grow Pinot Noir and Chardonnay wasn't in California, but it was actually in the North Willamette. And so Chuck Corey, that's Chuck, there's his nursery that he had with Dick Erath for a period of time. He he decided he wanted to come to Oregon. Well, his wife, Shirley, was fluent in French. And so right after he graduated from UC Davis, he got an internship at the French experiment station at Colmar in France. And when he was there studying grapevines, they were doing trials on these clones. And so he was the first one in Oregon to do something he shouldn't have done. But you can understand his frustration, but still. Well, the federal law in I think it was 1946, the federal government prohibited any importation of plant material that did not go through a quarantine process that was properly certified. And so, but the federal government then, of course, didn't fund that any kind of research or any kind of certification process. And so basically all the legal importation of plant material in the United States came to a halt. And so what Chuck Corey did was he brought with him on his trip back what they call the Corey clone. It's really the first suitcase clone that we call it, or Samsonite clone is another term, back into Oregon. And there's a danger, of course, and that would be that clone had virus in it. Of course, that experiment station was doing these studies on plant material. So I'm guessing that he did not bring any. He knew what he was doing, but it was still a risk. That clone we think now was clone number 538, which has actually been cleaned up by UC Davis and available from the plant service there. Here you can see Bill Fuller in Virginia. And this Bill Fuller, he was the winemaker at Louis Martini. In fact, he was the only non-family member of the Martini family that was making wine at the winery. And they actually encouraged him to take classes at UC Davis. And that's, of course, where he encountered Richard Sommer and David Lett. Next to Bill is Bill's first wife, Virginia. And this is a great story. The Virginia actually, in the backyard of their house in St. Helena, helped David Lett cut up the sticks, the cuttings, so they could be transported back with David Lett up to Oregon in a horse trailer that David Lett had borrowed from his uncle. Anyway, I just thought I'd share that fun story with you about in the role that they played. Now, Bill Fuller, when he came up planted in 1974. He planted the Draper Clone of Chardonnay, and you'll see this. This is the Draper Clone. If you look really carefully on the left-hand side, you'll see moss growing on this this vine. That's one of those really old vines at 12th and Estate. You know, the trunks are this big around. And that is the original Draper Clone. And because of when he brought it up, it did not go through a heat treatment. He got it from the Draper Vineyard, which was just behind basically where Virginia and Bill lived. And that clone actually came from Stony Hill, which originally came from Wente, and Wente got his Chardonnay clones back, in the late 1800s from the family did from France. And so that's another kind of introduction. Now, you think about, well, how does this clone perform? Well, here's something to think about. Bill Fuller was the very first Oregon winemaker to place an Oregon wine in Wine Spectator's top 100 ranking. And he did it with Chardonnay. And Bill Fuller is the only winemaker in the world to have won Best to Show White and Best to Show Red in the same year at the London International Wine Competition, where the trophies were presented by the Queen. How about that? People don't know that story. And anyway, Bill has given the trophy to us, and the trophy for the Pinot Noir is in our tasting room in the estate. Now, the dean, I would say, of the entire wine industry is David Adelstein. David Adelstein, even though David Adelstein was an intern in Burgundy. He left, he went to, as an intern to Burgundy as well, near Beaune, and he actually interned at one of these research vineyards that was doing these clonal studies. So to give you a little bit of history, of course, Corey went in 1964. David Lett actually went to Burgundy in 1965, encountering these clonal experiments. And then David Lett in 1970, David Adelstein in 1974. And David's aha moment was when he was there, he said, well, good heavens, he says the Chardonnay is ripening at the same time as the Pinot Noir. It doesn't happen that way in Oregon. And because Oregonians were having a terrible time getting their Chardonnay ripe. Well, remember that was heat treated. And so the 108 clone was of Chardonnay that came up from UC Davis, was heat treated. Now the Californians loved that 108 clone because it ripened later and it yielded heavier than the clonal material before it was heat treated. We didn't know that at the time. In fact, very few people know that now, that that's really what happened. Now, David Adelstein played a big role with Bill Fuller and others. Bill Fuller actually was the president of the Oregon Wine Growers Association, I think 3 times, providing leadership in encouraging Oregon State University to import this plant material. But David Adelstein was really the organizer and the dean of the wine industry then, and actually, I think now. He's really my thought leader in Oregon wine. And one of the things that he did was he went before the Oregon Liquor Control Commission in 1977 and got truth in labeling laws passed into Oregon law, which means that you couldn't misuse the geographic names of other wine regions like Champagne or Burgundy or Chablis. You had to call the wine by its true varietal name, and you had to, if you used a geographic designation, it had to be truthful and accurate, truth in labeling laws. Well, when the French saw what David and others had done in Oregon law, they were receptive to our interest in seeking their help in getting the original plant material from Burgundy. Now, what David and others talked, a plant pathologist, Ron Cameron, in this picture, Ron Cameron is the Oregon State University's plant pathologist, and he applied for and got a federal permit to import plant material legally into United States and into Oregon in 1975. And that's when things began to happen for Oregon, because the wine growers, the Oregon wine growers couldn't talk the Californians into using their permit to bring those plant materials in. And so they felt, well, like, if we can't talk the Californians to do it, let's just do it ourselves. And so that's what Ron Cameron made that application. And then along came a fellow named David Heatherbill. He was an enologist at Oregon State University, and he had actually spent some time in Burgundy and knew the researchers in Burgundy, and so played a big role in making sure we got that plant material. So here's some of the clones that the enologist Heatherbill was able to talk the French into sending us. And here's these, you can see in the clusters, the difference in their shape. And you can see how easily it was to classify these particular clusters. And so in the first group of clones came clones like 114, 115, 113 Pinot Noir, and then the 96 and 97 Chardonnay clones. Prior to that time, the earliest clone to come into Oregon came from an experiment station. And this is where David Adelstein discovered a Chardonnay clone at the experimentation called Espaguette, which is actually down near the Mediterranean in the Southern Rhone, but that's where the research station was. It was a Champagne clone of Chardonnay. And David Adelstein said, boy, that number 352 Chardonnay could make a really good sparkling wine. And so it was on David's recommendation that I planted the Espaguette 352 in our estate vineyard to the left as you come up the hill to the winery. And here's some examples, some other clones, and you can see the different clones, the 76, the 95, and the 96. So, and you can see the differences in their shapes. And they all have different flavor and aroma profiles besides being different in shape. Now, one of the things I got to do was I got to go with David and with the governor on the governor's trade mission to Burgundy to talk the French, to thank the French for those earlier shipments of plant material, but also to ask them if they would please send us more. And that was that 667 and the 777 clones. So you see me here, pretty young in this picture, and actually dressed as a member of the trade mission in a vineyard just outside of Beaune. And here are the clusters that resulted from the plant material that came, the Dijon 667 and 777. This next slide, you can see some of the detail. And so I'm going to spend a little bit more time on this slide because there's a little bit more detail here. Obviously, you won't be able to digest this all at once, but you can see these different clone numbers, and then you can see their style, the fragrance, the aroma, their body, their tannins, and you can see how different they are amongst these different clones. Now, the most popular clone amongst that first set was 115. In fact, there's for those of you who like the henna that we produce, that predominantly comes from a planting of 115 at the crown of the hill at the Peter Michael Vineyard by 12th and State. And then the 667 and the 777 have also very unique qualities. The 667 has got this holiday, spice, and the 777 has got, these wonderful kind of cherry, blackberry and tobacco aromas and flavors, but they're very different. And actually, many people don't know that Robert Drouhin, when he decided to plant a vineyard and build a winery in Oregon, which he announced in 1988, he actually encouraged us to appeal, to ask for the 667 and the 777. So he knew what was the right thing to do. Well, I was in such a hurry when we got this plant material that I ran out into the vineyard and we actually field grafted these vines. And so you can see how we took the little, budwood, and we used a tape, and we taped that little piece onto a niche that we cut into the original vine, which was actually Riesling in that block. And we grew, because I didn't want to wait. I wanted to make sure we got the 667 and the 777 growing as fast as we can. And of course, you now know that what was the penalty for that. And if you stand on the deck and you look down into that block, you'll see in the 777, there's kind of looks like a big hole in the vineyard. And that's because the original planting of Riesling had phylloxera on its roots. And it finally took hold and it's finally killed those vines. And so those vines are now being replaced with phylloxera-resistant rootstock. Now, I wanted to make sure that you understood just how vines behave. And so this next image is an image of a UV light. And the reason why I use this example is it is Earth Day after all. And as you know, we're dedicated to being as sustainable as we possibly can be in growing our grapes, making our wine. And so I wanted to explain to you just how, now think about this. We and other mammals can move around. So when the environment changes, we can kind of figure out how to go someplace else. Plants can't, they're stuck. And so plants are far more adaptive to environmental conditions than mammals. They're actually designed that way. And we actually, the plants and other things like powdery mildew or other things, viruses and the like that affect plants are actually highly adaptable and mutatable. That's why there are probably hundreds of different mutations of Pinot Noir. Well, what we're doing is we're applying UVC light on powdery mildew at night because powdery mildew has a repair mechanism in its DNA that has developed over time. And of course, powdery mildew knows that the sun goes down. So it shuts off its repair mechanism when the sun goes down because there's no more UVC light. By applying UVC light, which can be damaging to the cellular and the genetic structure of powdery mildew, if we apply that at night when the repair mechanism is shut down, the powdery mildew can't repair itself, and so it dies. And so this is a way of killing powdery mildew, which requires organic sulfur, which is what we spray on the vines frequently, or it requires many farmers to apply a fungicide, which really isn't good really in large quantities for the environment or for us or other living creatures. So we are actually leading an effort to develop a method by applying this UV light at night as a way of killing powdery mildew, but also eliminating the harmful chemicals that can be applied in vineyards and in agriculture. Well, I wanted to show you this person. She's actually, you see a lot of men in these slides. Well, this is a remarkable woman. Her name is Frédérique Pellegny, and she is the head of research at the experiment station in Colmar in France. She's a research scientist. She's been working for 15 years to unlock the mystery of why do we have so many different clones? Where are they mutating? Why are they mutating? And why do we get the results we do? After 15 years of research with her team, it was only 6 years ago that she was able to prove. And here you're gonna be able to see this on this next slide. You're gonna see a cutaway of the grapes. And there's obviously not enough time to study this slide, but I want you to pay attention. You can see the skin there where the anthocyanins are located. That's what those genes produce, the color. And you can see the vascular that's right underneath that cutaway. And in the next slide, you're going to see even a more detailed image of that skin. So you can see that there's this kind of waxy surface on the surface, and then you go down further, and you can see these layers of cells. Well, what Frédérique she discovered is that, and her team, that there's a place on the gene that if you have two genes that simultaneously mutate, they will turn off the instructions to make anthocyanins that are responsible for color in the grape cluster. So that's why you have Pinot Gris from Pinot Noir, and you have Pinot Blanc from Pinot Gris. The other thing that she discovered, which is mind-blowing, and it's hard to understand this or think about this probably, but once it does make a change in one berry, that signaling will actually spread to other berries. And that signaling, that genetic mutation will actually signal and spread to other clusters. That's why you can end up with Pinot Blanc, for example, on a fruiting cane that is from a Pinot Gris vine, or Pinot Gris from a Pinot Noir vine. It's fascinating. And so all you have to do is just cut off that cane, that 1-year-old wood, nursery it, and now you've got a new variety. And that's where it came from. And so you can see, this is a little bit more detailed. You can see here of the chromosome, the gene, and you can see these mechanisms and some of them she discovered are more adaptive, kind of mutation sensitive than others. And which leads to the differences in the clones. So even though the Pinot Noir skin doesn't change color, you get different aromatics and flavors from as a result of berry because of those mutations that occur. Now, there's a scientist in Australia that here this fellow's name is Dr. Andrew Gearing. He's a plant pathologist at the University of Queensland in Australia. Because remember, we're still digging deeper. We're trying to figure out, where did Pinot Noir come from? And he found it. And he found it in what's called the intron part of the gene. He found a fragment of basically a prehistoric virus. And this prehistoric virus, they estimate, is like 34 million years old. And he believes he's traced back this virus to about 1.8 million years ago, that this fragment of virus altered the grape and produced Pinot Noir. So he believes that 1.8 million years ago, Pinot Noir was the very first vinifera grape. And he figured out, he traced it back, and he thinks it's from the intron here. And so anyway, I just thought I'd, what he explains is that he says, when you have a mutation, whether it's UVC light or some other environmental condition that damages these genes, these repair mechanisms are basically like, it's just like basically putty knives, and they're basically grabbing material, genetic material, where they can to fill in the damage that's been done to the gene and the chromosome. Well, they basically have taken some of that material of this virus, and they've basically filled the crack in the gene, almost like with a putty knife. That was the repair mechanism. That's how that plant changed to Pinot Noir. When you come and visit me at the winery, you're going to see what's called the Bernau block. And what I'd like to do is invite you, when we have an opportunity, to go and walk along the top of these rows, because on the left, I have Pommard growing that I got from Susan Sokelblosser. She actually even helped me lay out the vine rows. And then these middle vines are Wadensville clone. And I got that from the Castiles at Bethel Heights. And then we have the 667 and the 777 clone that we got from the winemakers who generously provided that plant material to Oregon State, went through quarantine, and then was released to us. In this image of the estate, this is kind of an image of the layout of the vines. You can see if you this is kind of a topographical image. So it's hard to know the roads. You can see Enchanted Way enters there on the far left at the top, makes its way up to the estate tasting room. And you can see the blocks here. You can see the Chardonnay that's planted. And then you can see the Pinot Noir blocks that are planted in red. Well, I wanted to tell you that there's a block up near the house, right below my house. And there's a clone that I got called 943. Now, this clone, what happened is eventually the French figured out, oh, we better start licensing these clones and charging for them. So they trademarked them and licensed them. And of course, they do certify that they're 100% virus free, but they charge 30 cents each just for the privilege, just the royalty privilege for buying each plant from a nursery. And there's 3 nurseries in the United States that have a contract with the French to produce this plant material. So I had to sign a licensing agreement that says that I can't let anybody take cuttings. I can't take cuttings myself from this 943. But that happens to be Dr. Raymond Bernard's favorite Pinot Noir clone. Before this clone, his favorite was 115. This next image is of Ingram Estate, which is just above Elton. So you can see Elton in the top right. That's Elton Vineyard. Further down the slope in the Eola Hills, as you come up the slope, that's Ingram. That's our property that we own. And here we have a variety of plantings. So this next image, you'll be able to see these plantings. You'll see both Elton and you'll see Ingram. Now look at all the Pinot Noir. See all that red that's planted? Pinot Noir. The pink is the Chardonnay. And then of course you see a little blue, which is Pinot Gris. Now, I got to tell you that in this block, for any of those of you who are with me, brought your shovels to plant Pinot Noir vines, we planted, this was back when we started this vineyard. We started the Elton brand, remember that? And you came and helped me. We had over 500 people helping us plant Pinot Noir vines with their own shovels in what's called the Fluorine Block. And the Fluorine Block is in this middle section of this image. And I got to tell you, we've got a couple other clones in there besides the 113, 114, 115, and the Pommard and Wadensville. We have 2 other clones that are referred to as, one of them is referred to as a heritage clone, which is really kind of misnamed. Because remember I told you about these suitcase clones? Well, and I told you how some clones had the names of the vineyard owners rather than numbers. Well, there's a clone we planted here called Swan. And we got Swan actually from UC Davis. UC Davis has properly certified this and made sure it was virus-free. They call it a heritage clone, but actually it arrived in the United States long after the it was legal to import, and we had locations where you could import vines. The person's name who brought this plant material was a real Burgundy nut who was also an airplane pilot for Western Airlines. So you can imagine how he got the plant material into the United States. Well, it would turn out to be such an interesting Pinot Noir clone that UC Davis included it in their heritage clone category, even though it got imported in the United States in the 1970s, well past the time we were actually legally getting plant material. But I thought you'd get a kick out of that. The other thing that's planted here is called Mariafeld. Mariafeld is from Switzerland, just like Espaget is from the experiment station there in, pardon me, not Espaget, Wädenswil Pinot Noir is from the Wädenswil experiment station, Swiss experiment station there at Lake Zurich. And this Mariafeld, what it does is it has very loose berries. And the berries are a little bit larger than those smaller berries you see in Permard 115, 114, 113. So looser and larger. And the reason why this clone is valuable is because it's fairly high acid, and it has big berries, so it's perfect for sparkling wine production. And the other reason why it's an advantage is because these loose berries means it's less susceptible to botrytis, because botrytis, that rot, right, that gray mold, really affects the tighter knit berries a lot more than the looser berries, because the air can dry out the water between each cluster. Now, this image here you see is of Tualatin. Many of you will recognize this vineyard, planted in 1973 by Bill Fuller. There's the tasting room, which is really a converted strawberry processing shed. And in this next image, you'll see that this has really a very diverse planting where there's a great deal of commitment to Pinot Noir, as you can tell in this. But I wanted to call your attention to, we have really a couple of treasures here. I mean, just real treasures, because we have heritage clones that are the real McCoy, the very original plant material. So you see down here, there's some pink, and that's actually right to the below the house that Efren lives in, where Bill Fuller used to live years ago. That's called the Draper Clone. It's planted on its own roots. It has not been subjected to any treatment of any kind. It came up from the Draper Vineyard with Bill Fuller, and that's that very special Chardonnay clone. I hope we can keep those vines alive, because this is like living history. And then also there's a Permard planting further up the hill that is also very unique and old planting. So this next slide shows you the Peter Michael block of Tualatin Estate. This gorgeous picture. And in this next slide, you can see the layout. And in this layout, what you can see is we have done essentially an incredible experiment, because you can see all these different Pinot Noir clones, and you can see all these different rootstocks that are listed with these clones. And there you can see, if you look close in that upper right-hand portion, you can find the 115 plantings of those of the Hannah fans that are on this Zoom. But we have, in a way, our own little form of dealing with a concern that the French winemakers had about being homogeneous with plantings of clones. They are still convinced of the importance of the Masala approach, of genetic diversity in the vineyard, because they believe that that produces long-term more genetic diversity among the vines as they change over time, responding to environmental conditions and mutating. And they believe that that's a better approach. And so what we've done, in a way, is we've got our own kind of large-scale Masala. This last image is an image of what we're building in the Orville Roth barrel room underneath the estate room. We're creating an experience for wine enthusiasts to come and learn about how these different clones, what their aroma is and how they taste. So each barrel will have 113, 114, 115. It'll have 667, 777. It'll have Permard and Wädenswil Pinot Noir clones in each of the separate barrels, and consumers will be able to come and use this very special system that David Markell, our director of research, is building, that allows you to calibrate the amounts that you want in your taste. So you can experiment, you can learn about each clone, but then you can experiment to see what happens in the combination. Because here's the thing that you'll discover, and I don't know if any of you are on this Zoom that have done this with us, because we use pipettes and graduated cylinders now. It takes a lot longer, and you've got to have a calculator and a lot of paper. But this will do it. This will do it automatically as you make those adjustments on the dial. And so it'll be much easier to do and much more accurate, actually. But what you're going to learn is this. You're going to learn that when you put these clones together that have these different aromas and flavors, but you put them together in particular combinations, you're going to get even different aromas and different flavors. Now that's the mind-blowing experiment that I want you to do. David Markell says he thinks you'll have it operating by Mother's Day. We'll see, but I don't think you'll find anything in the world like what David Markell is building. It's actually very similar to the barrel blending system we have in Folsom that blends varietals at the Natoma barrel blending system at our Willamette Wine Works in Folsom, California. Anyway, that's what I wanted to share with you in the terms of the slides. Look at the time. It's 59 minutes, and I actually beat Christine's bet. She thought I'd go 6 minutes longer. So, but anyway, I hope we've got some questions. She's going to ask me some questions. If you're willing to stay on the Zoom to hear some of the answers.
Jim, it seems like you did a very, very thorough job, as we don't have any outstanding questions, but we can give guests a few moments if they do have a question to answer or to ask it now, and for you to answer.
Yeah, I wanted to remind everybody, my email address is my name, jim.bernau, B-E-R-N-A-U, @wvb.com. The contest is open through the end of this evening, all the way up till midnight, where you can tell me, based upon the information you've derived in this presentation, that clone, that first Pinot Noir clone we showed you, what does it most like? Based upon the descriptions that I gave, what is this Pinot Noir clone most like of all the descriptions that I gave? And there should be an answer in this presentation. So we'll see how you do.
Wonderful. Well, all the questions have been answered, and Jim, thank you so much for leading this history and explanation of the story that's not widely known. We do have a new question, sorry. So given that it is Earth Day, any thoughts of how climate is impacting the vineyards and wine grapes?
Well, we know it is. All of our farming records show that temperatures are increasing, the plants are behaving differently. A couple of things that are happening, obviously, is that the maturation time, and harvest is coming closer, to us. In other words, it's occurring earlier. We've also found that the growing season has shifted a little bit into the fall as well. And we found that there can be danger from heat spikes. So there's more inconsistency in the weather over the growing season, and more fluctuations that are more extreme. And the danger there is what happened, of course, in some places in the United States where frost, killed their shoots. And that's happened actually in Burgundy and in Champagne this year, where they had historical frosts. And so you have these weather patterns that are more extreme. The other side of it is to have heat spikes. Now, what happens? Pinot Noir is so sensitive a grape that it has a growing tolerance that's very narrow. So believe it or not, as the temperature gets warmer, above about 98 degrees, the metabolism in the berry slows down, and it can actually stop if it gets too warm. And then it's being called sunstruck. It actually will stop permanently. So we actually, in periods of time when we have real heat spikes in some years, we have to go remove those green berries that didn't properly mature from the cluster before we can make the wine. So those are a couple of issues that I think should be a concern to you. The thing you probably know by this presentation is that early on, the French were interested in figuring out how to get grapes to ripen earlier by doing clonal selection. Well, now you've also learned that by doing clonal selection, we can get grapes to ripen later. So there's actually a solution besides just moving up higher in elevation on our slopes. There's another solution, and that is clonal selection to dealing with this warming conditions that we have. The best thing we can do is we can do everything we can as individuals, right, to reduce our impact on the environment. And one of the ways you can do that, it's very simple. Choose wine enclosed with natural cork. You can reduce the carbon footprint of a bottle of wine by 25% just by choosing natural cork as its enclosure, as opposed to using a plastic or aluminum screw cap.
Jim, we have another question that came in from Joe Higgins. He wants to know, out of all these clones that you've described today, what is your favorite?
It's that's tough. It's hard to say because each vintage year, these clones will express themselves differently. Even in each vineyard, these clones express themselves differently. So that's really hard to say. But I can't wait, actually, until the 943 clone we have sufficient crop of it for us to make that as a separate bottling. And I'll tell you what, let's make sure he's there with me when we pop that bottle open and we taste a 100% 943 to find out what Dr. Raymond Bernard was talking about. Now, this 943 cluster is really small. One of the reasons why a lot of people don't favor it is because it's hard to make any money growing it. It's really small. The berries are really small, but it has this incredibly intense flavors and aromas that come from it. But you don't see it very frequently planted because it's you have to charge a lot of money just to cover the cost of making the wine.
We did have another question. Don was asking, how would you describe the 2019 Dijon Clone Pinot Noir?
Oh, wow. Well, I think I might have given a few clues to the wine. I would say that it's really lively. That's one thing I would say. In other words, I think this has got a long cellar life. It's got a blade of acidity, which makes it perfect for foods that require that. So it's very vibrant. I think it's juicy and silky. So I think you're going to find a real strong potential in aging with this wine, and it'll get even like satin over time. I do like, kind of that earthiness that's in the wine, and also the expression of the various kinds of, kind of aromas that you get, whether it's raspberry or cranberry, for example. Remember, it's still a young wine, but if you, like you do what my wife does, make some chicken Marbella and have it with that.
Great. Well, that concludes all the questions. We did have a request to send out the recording so people can rewatch to pick up on things again and again. So we will be sure to send that out to everyone that registered today and joined us.
I took some risk, Christine, in I presented quite a bit of material, and I took a little bit of a risk in kind of the abridged version, and I didn't include a lot of technical information because I knew that that's not really what the staff really needed to understand the value of these clones. And I also tried to figure out, I mixed in some stories because I wanted to make sure I kept the story alive. It's wine is like, drinkable history. And so I wanted to take a subject that could be actually fairly dry and bring it alive. And I hope I've done that for all of you today. And you can see that that it's really been the combination of many, many people who created the Oregon wine industry, that created these opportunities for us. And I just highlighted a few, and there are many, many more that could be, the contributions are very worthwhile in creating these opportunities, clonal opportunities for us. So I just touched on it. So I hope you'll give me a little bit of license in how you view the presentation today. Thank you all very much. You have a great Earth Day evening and dinner. Thank you all for joining me.